AS Level Biology 9700, Paper 1, Variant 2, October November 2024. And question number 1 is, a light microscope is used to observe two structures that are 200 nanometer apart on the slide. So these two structures are present on the slide, which are 200 nanometer apart. So what is the actual distance between the two structures when the magnification is changed from 40 to 400? So students, you can say it, or the distance is shown on the slide. So when we increase the magnification, so only the magnification is increased. There will be no change in the actual distance between the two structures. So it will remain the same, that is 200 nanometer. So according to this, C is the correct option. A, B, and D are not correct because there will be no change in the actual distance on slide when we increase the magnification. Now the question number 2 is, a cell is shown in the micrograph. Which statement explains how it is possible to identify the type of microscope used to produce the micrograph? You can see nucleus is shown, endoplasmic reticulum are there, and you can see a dot-like structure on the endoplasmic reticulum, which are ribosomes. So these different organelles are only visible under the electron microscope. Light microscope only on the nucleus. So the nucleus, the first option is the nucleus is visible, so an electron microscope was used. No, it is also visible in light microscope. This is not correct option. The endoplasmic reticulum is not visible. We can see endoplasmic reticulum is there. So correct option, the chloroplasts are visible, so a light microscope was used. In light microscope, we cannot see the chloroplasts, ribosomes are visible, so an electron microscope was used. Yes, this is the photograph which has been taken from the electron micrograph, and ribosomes are visible on endoplasmic reticulum. You can see the dot-like structures on endoplasmic reticulum. So D is the correct option. Now we are moving towards the question number 3. And question number 3 is, which statement which statement supports the fact that mature plant cells contain organelles that carry out the same role as lysosomes? And what are the lysosomes? Lysosomes are the organelles in animal cells that break down waste and recycle materials. So they contain digestive enzymes that break down proteins, nucleic acid, lipids, and carbohydrates. So the first statement is, a range of hydrolytic enzymes can be found within mature plant vacuoles. Yes, it is correct. That is, the hydrolytic enzymes are present in the plant cells, in mature plant vacuoles, which contain hydrolytic enzymes. So these enzymes break down cellular materials which are not needed. So in this way, we can say that this statement can be the correct. Second statement, glycogen found within vesicles can be hydrolyzed to glucose molecules. This is the breakdown of glycogen into glucose molecules. When the water is added into the glycogen molecules, then they will break down into glucose molecules. So the double membrane bond vesicles are formed from plant Golgi bodies, double membrane bound vesicles. So from the plant Golgi bodies again. This is not the correct option. So the vesicles formed from the cell surface membrane contain enzymes. So vesicles which are formed from the cell surface membrane, they contain enzymes. They are the lysosomes, which are formed from the cell surface membrane, contain enzymes. But these vesicles or the lysosomes, they are not present in the plant cells. And we are talking about mature plant cells. Again, the option two, glycogen. Glycogen is also not stored in plant cells. Glycogen is stored in animal cells. Double membrane bound vesicles are formed from plant Golgi bodies. So in this way, the again not correct option, and D is not correct option because these are the lysosomes. So only A is the correct option, according to which there are in the mature plants there are hydrolytic enzymes uh, which can break down the substance or the material which is waste and not needed by the plant. So, you know, the vacuoles are storage, just like the storage bubbles in plant cells. So that perform many functions, including storing nutrients and toxic materials. So these hydrolytic enzymes, they break down the macromolecules like proteins, nucleic acids, and polysaccharides. And same is the case in the animals which is performed by the lysosomes. So here in this case, A is the correct option. Now we are moving towards the question number 4. And you can see the question number 4. Which cell structures may contain cisternae? So here you can see the chloroplast and endoplasmic reticulum, Golgi bodies, and mitochondria. So, cisternes. So, cisternes are flattened sac-like structures that are part of the endoplasmic reticulum and Golgi apparatus in the cells. They are membrane bound and play a key role in the cell's packing, modification, and transport of proteins. And you know, these cisternae are only present in endoplasmic reticulum and Golgi bodies. You know, the mitochondrion and chloroplast. All they are double membrane structures, but they don't contain these cisternae. So only they are present in endoplasmic reticulum and Golgi body. So according to them, the C is the correct option for the for the question 4. Now we are moving towards the next question, and next question number 5 is the diagram shows some cell structures of one type of cell. Here I think this is the prokaryotic cell. So which labeled cell structures are present in typical eukaryotic cell and a typical bacterial cell? Here they have shown a bacterial cell, and at number one there is cell wall. At number 2 there is capsule, flying capsule. At number 3 is DNA or hereditary material. Number 4 is ribosome. And number 5, you can see there is cytoplasm. So it means you can see the eukaryotic cells contain cell wall, they contain hereditary material, they contain ribosomes, they contain cytoplasm. So they don't contain this slime capsule. So B is the, in this case, you can see the B is the correct option. Now we are moving towards the next question. And next question is 6. The diagram shows part of the collagen fibril made of collagen triple helix. The collagen triple helix are linked to each other by one type of bond. This bond is labeled as X in the diagram. So you can see these 3 collagen helix. So they are called as triple helix because they are 1, 2, 3. So what is bond X? So what is bond X? The main, you can see these are these cross links. These are the cross links which are present between these three helicals. They hold tight together. So these collagen fibers, uh, you can say, held together by these cross links. And students, these three helicals uh, are called as triple helical helix. So what is bond X? So the bond X, these are the cross links, and this is the covalent bond A. And uh, these covalent bonds, uh, simply we can say these are the cross links. So these cross links provide the collagen fibrils with their tensile strength and stability. So tensile strength and stability, uh, only these covalent provide, whereas the disulfide bonds are important. We can say they are also important in stabilizing protein structures, but they are not primary type of bond involved in linking collagen triple helix. So these disulfide bonds exist in collagen fibrils, but they are not uh, work as crosslinks. And third option is the hydrogen bonds. Hydrogen bonds are present between the collagen molecules. So again, this is not the and peptide bonds are the also the covalent bonds. They join amino acids together to form proteins. So they are present within the collagen triple helicals. They are not the bonds that link the helical together in the fiber. So in this way, only A is the correct option, which is covalent bond. And all they are not the correct options. They exist, but they are not performing the function. That is, they are they are not the cross links between these three helicals. Question number 7. And question number 7 is, the table shows some information about the polypeptides that make up hemoglobin. And here you can see that they have given us a table, and this table shows that total number of amino acid residues in polypeptide chain in alpha globulin is 141, and beta globulin it is 146. And then position of amino acid cysteine in polypeptide chain is 104 and 93 and 112 in beta globulin. So here you can see the alpha globin and the beta globin. Protein in red blood cells. Hemoglobin, what is hemoglobin? A protein in red blood cells. So they consist of 4 chains. They are the 2 alpha globins and 2 beta chains, alpha chains and beta chains. So scientists studied the region of the beta globin polypeptide chains containing the amino acid cysteine at position 93. So they found that this region faces outwards when no oxygen is attached to the heme group. This region faces inwards when oxygen is attached to the heme group, replacing cysteine with the different amino acid residues, the Woher shift. So according to these 3 statements, they have given us 4 options in which we have to conclude from the information about cysteine in hemoglobin. So according to this, we can read one by one. That is the, what is, first of all, we should know what is cysteine. Amino acid that can form disulfide bonds in protein structure. So cysteine is present in the structure of hemoglobin. So what is Bohr shift? Next, the effect of pH on the oxygen affinity of hemoglobin. Lower pH decreases oxygen affinity facilitating oxygen release in tissue. So first of all, we are going with the question statement A. More than 1% of amino acids in one hemoglobin protein are cysteine. Yes, this is correct. This option is correct because after every third, you can say the amino acid is the cysteine. So next is in the alpha globin, there is a cysteine closer to the end of the polypeptide chain with an unreacted carboxyl group that in beta globin. According to this, this option is not correct. Okay? Because the table only shows us the positions of cysteine, it does not tell us which end of the chain the positions are closer to, nor does it mention anything about carboxyl groups reacting or not. So this is not the correct option. Again, we come to the point 6. Uh, the, re the replacement of the cysteine at position 93 in beta globin decreases the affinity of hemoglobin for oxygen at low pH. So according to this, this is actually related to the change in the oxygen affinity at different pH levels. So if the Bohr shift is reduced, the difference in oxygen affinity between high and low pH is smaller. So this means that the hemoglobin with the cysteine replacement will have a higher oxygen affinity uh, we have higher oxygen affinity at low pH. So again, this is not the correct option. The binding of the oxygen to the heme group causes the region of beta globin containing cysteine at position 33 to become more hydrophilic. So this binding of oxygen to the heme group causes the uh, this is causes the region of beta globin containing cysteine at position become more hydrophilic is again a wrong option. This is not correct option because this information provided leads us to conclude that the cysteine at position 93 is involved in the pH dependent oxygen binding of hemoglobin. So according to this, replacing this is lessens the pH effect. So we can see that this is not the correct option. Only A is the correct option in this case. Now we are moving towards the question number 8. Yes, we are on question number 8. Which features of glycogen distinguish it from starch? Glycogen and starch. All glycogen molecules are highly branched, and starch is more branched than the glycogen molecule, as starch is made up of two types of structure, amylo and amylopectin. And there is formation of one four and glycosidic bond and one six glycosidic bond in starch. So starch is made up of amylose and amylopectin. And amylopectin. So these are the two types. So according to this, the all glycogen molecules are polysaccharide. Starch is also polysaccharide. All glycogen molecules contain alpha glucose. Same. The all glycogen molecules contain wide bond glycosidic bond. So only A is the correct option in this case. Glycogen is highly branched and starch is also branch structure, but it is made up of two different molecules as amylose and amylopectin. So they are different from one another, although they are both of they are the polysaccharide molecules, but they are different from one another. So the answer is A. Now we are moving towards the question number 9. And question number 9 is the diagram shows a biological molecule. And here you can see the diagram shows a biological molecules and which molecule which molecule would be produced if this biological molecule was hydrolyzed. Here you can see they have shown the triglycerides. When they are broken down, they are changed into glycerol and fatty acids. So when they are hydrolyzed, they are changed into fatty acid and glycerol only. Fatty acids, glycerol, and these are the three fatty acids. So C is the correct option. And what is A? Amino acid and glycerol only? No. Amino acids, these are not amino acids, these are fatty acids. So not the fatty acids, glycerol and water. Water is not produced during this, so only fatty acid and glycerol. So 9, C is the correct option. Water is not produced, but you know the water is added into it. They are broken down there. Well, the question number 10 is a mixture of glucose and starch solutions was placed in a length of dialysis whisking tubing and the tubing sealed. The tubing was then placed in a boiling tube containing distilled water. Two samples were immediately removed from this water and tested with either iodine solution or Benedict solution. You know, the iodine solution is used to identify the starch, and whereas the Benedict solution is used to identify the presence of glucose in a solution. So this was repeated at 10 minutes interval for 30 minutes, for half an hour, you know. So the iodine solution gave an orange brown color each time, iodine solution. So the table shows the results of the Benedict test. So at 0, it was color produced by Benedict test was 0. At and then at 0 time, when there was no reaction, but when the reaction started, it gave, first of all, it gave green color, green to yellow, yellow to red. So it is a complete sequence when Benedict test is performed into the laboratory. So it shows the presence of Benedict solution. So what may be concluded from these results? The pores in the whisking tubing are too small for starch tube to pass through. So according to this is correct option because uh, the starch molecules cannot pass through this whisking tube. So the glucose diffuses through the whisking tubing down our diffusion gradient. So that is the reason the Benedict solution, Benedict test is positive. That is the red color shows that the Benedict test is positive and the glucose was present there. So it means glucose diffuses through the whisking tubing down a diffusion gradient. So this statement is also correct. Water diffuses into the whisking tubing. Whisking tubing, no, it does not happen. It is not diffused into the whisking tubing. So this is not the correct statement. So according to these two correct statements, the answer is B. Now we are moving towards the question number 11, and question number 11 is there. You can see which molecules are globular proteins. Amylase is an enzyme which is a globular protein. Hemoglobin is a red protein which is present in the red blood cells. It also a globular protein. DNA polymerase is an enzyme uh, for the synthesis of DNA or protein synthesis. It is also the globular protein. So according to this, these three are A is the correct option. Now question number 12. And question number 12 is, you can see over here, the initial rate of a reaction catalyzed an enzyme was measured at various substrate concentrations. This graph shows the effect of a low concentration of non-competitive inhibitor on the reaction. Students, you know the competitive inhibitor, it will try to bind on the active side of the enzyme, whereas the non-competitive inhibitor, it will not bind on active side, can bind anywhere on the enzyme. So it will affect the reaction. So we have to find out which is the correct option. According now the graph A, initial rate, you can say the substrate concentration, and the dotted line shows the without inhibition, and the straight line shows with inhibitor. So you can see it shows the process taking place. We have to find out the non-competitive inhibitor. Then the non-competitive inhibitor binds with the enzyme on any place. It change the shape of the active site there, due to which there will be no reaction takes place. So, but you can see a reaction is taking place over here. So this is not the correct option. Second B, again the reaction is taking place. In C, the reaction is also taking place. But here you can see on D, a low concentration of non-competitive. Low concentration means the reaction started, then it stopped. After some time, it stopped. It means D is the correct option. Because I have already told you that when there is non-competitive inhibitor, it will bind anywhere on the enzyme, and then it will change the shape of the active site, and then due to which the reaction will not proceed. So D is the correct option in this case. Now we are moving towards the question number 13. So question number 13 is, you can see gout is a type of arthritis. Gout is a type of arthritis, so in which small uric acid crystals form inside and around the joints. This causes sudden attacks of severe pain and swelling. So it can cause severe pain and swelling. And the diagram shows how uric acid is formed from hypoxanthine catalyzed by the enzyme xanthine oxidase. So here you can see in the uh, equation, xanthopene in the presence of xanthine oxidase is changed into xanthine. It changed into xanthine, and xanthine in again is changed into the uric acid. So gout can be treated using drug called allopurinol, which has a similar shape to hypoxanthine. So allopurinol. So it has this similar shape with hypoxanthine. And in the previous MCQ, I have told you that when there is competitive inhibition, then what will happen? The inhibitor will be of the same shape of the substrate molecule. So it will try to bind on the active site. So here in this case, as allopurinol has similar shape with the hypoxanthine, so it will try to bind itself on the active site. So when it will bind with the active site, so instead of formation of hypoxanthine, there will be the, you can say there will be no formation of uric acid. So it will help to reduce the formation of uric acid in this case. So according to this, we have to find out which one is the correct statement, or which one is the correct option over here. What can be it binds to the active site of xanthine oxidase instead of hypoxanthine, resulting in reduced production of uric acid. It can be a correct option because it is a competitive inhibition. So they will compete. Both of they will compete for the active site. So they will try. So this allopurinol, it will bind itself on the active site. So due to which there will be no formation of xanthine and uric acid. Only there will be the formation of, you can say, reduced formation of the uric acid. Secondly, it binds to another part of xanthine oxidase, and this changes the shape of the uh, active site. No, there will be, it will not bind because it is similar shape to the hypoxanthine. So this is a competitive inhibition, not a non-competitive inhibition. So the second statement shows the non-competitive inhibition. So this is not the correct option. It disrupts the hydrogen bonds within the xanthine oxidase and changes the uh, and shape of active site, and changes this denatures and the active site no longer complementary to hypoxanthine and xanthine. So we can say denaturation is a drastic process that usually involves significant changes in temperature or pH. So allopurinol does not cause such a dramatic change. So it does not explain the specific competitive mechanism suggested by the structural similarity. So this is not the correct option. Now the last option is there, which is D option. And according to D option, you can see that it hydrolyzes the peptide. It hydrolyzes the peptides bonds between xanthine oxidase to change the shape of the active site. Again, this is incorrect because hydrolyzing peptide bonds means breaking down the enzyme's protein structure. So this is also a form of denaturation, and this is far more destructive than the action of allopurinol. So it is not, we can say, the correct option. Only A is the correct option, in which they have shown the competitive inhibition because allopurinol and hyperxanthine xanthine are of having the similar shapes. So they will fight for active site. Now we are moving towards the next question. Question number 14 is there. Phospholipids. So the phospholipids are formed in a similar way to triglycerides. A sample contains 6 phospholipid molecules. The molecular weight of a phospholipid ion is 95 gram. The molecular weight of each individual fatty acid in this sample was found to be 282 gram. The molecular weight of glycerol is 92. The molecular weight of water is 18. What is the molecular weight in grams molecules? So we have to, the phospholipid molecules. Here, the phospholipid molecule consists of, glycerol and two fatty acid chains. So there are the 6 phospholipid molecules. So we have to calculate all these. And according to this, first of all, the phosphate is 1. So 95, but there are 6 molecules, so we will multiply 95 with 6, so it will be equal to 570. And then 282 fatty acids. So there are two fatty acid chains. So first of all, we will multiply 282 with 2, and then we will multiply it with 6. So after calculation, it is equal to 3384. And then there are glycerol. So 92 into 6. One glycerol and one phosphate molecule. So according to this, it will be equal to 552, and there is formation of 18 water molecules. So 18 multiplied by 6 will be equal to 108. So first of all, we have to add these. We have to add these, and according to their, let me calculate. So you can see over there, 570 plus 3384 plus 552 is equal to 4506. We have to minus the water molecules from it, because water, when there is formation of uh, phospholipid molecule, the water, this is a condensation reaction when the water is removed. So 18 water molecules will be removed. So when these 18 water molecules will be removed, then we have uh, 108. So we will mod 506 minus 108. First of all, we have to, and here I have to minus the 18 water molecules. So 18 multiplied by 18 will be equal to 324. So there will be 324. So we will minus 324 from 4505. Then 4506 minus 324. So the answer will be 4182. So according to this calculation, the answer is, for question number 14, is B. I explain again. Phospholipids are similar to the triglycerides. The phospholipid molecule consists of one phosphate molecule and one glycerol molecule. And there are two fatty acid chains. There are two fatty acid chains. So we will 95, there are 6 phospholipid molecules. So 95 will be multiplied with 6. 282 have 2 chains. So it will be multiplied with the 6. So on calculation is this. And the 92, 6 molecules, 1252. And there are 18, you can say 3 ester bonds. So 6 molecules are there. So there will be the formation of 324. So 4182.